The brain and spinal cord are enveloped by three membranes called meninges. The outermost membrane is the dura mater, the middle layer the arachnoid mater, and the innermost layer the pia mater. The meninges are important for the production, circulation, and reabsorption of cerebrospinal fluid. They also help to support the central nervous system and act as a mechanical barrier against infection. The dura mater, thick, strong, and inelastic, forms a strong protective sheath around the brain and spinal cord. Within the skull, the dura mater has two layers, an inner meningeal layer and an outer endosteal layer. They are closely opposed over most of their surface, but separate to enclose the venous sinuses. The endosteal layer is essentially the periosteum of the cranial bones. It adheres tightly to their internal surfaces, extending into the foramina and sutures as fibrous processes. The meningeal layer, the dura mater proper, forms tubular sheaths for the cranial nerves and fuses with the epineurium as the nerves emerge from the skull. It completely surrounds the optic nerve as far as the eyeball. Infoldings of the meningeal layer form septa, which partially separate the cranial cavity. The falx cerebri, the tentorium cerebelli, and the diaphragma celli. Inferiorly is the falx cerebelli. The falx cerebri lies in the midline between the cerebral hemispheres. It is attached to the crista galli of the ethmoid bone and the internal frontal crest anteriorly, and posteriorly to the internal occipital protuberance. Between the two, within its attachment to the roof of the skull, is the superior sagittal sinus. Anteriorly, the free lower border of the falx cerebri contains the inferior sagittal sinus, which drains into the straight sinus at the junction of the falx cerebri and the falx cerebelli. The posterior attachment of the falx cerebelli to the internal occipital crest encloses the occipital sinus. The falx cerebelli separates the cerebellar hemispheres. The tentorium cerebelli separates the occipital lobes from the cerebellum. It forms the roof of the posterior cranial fossa. Between the anterior clinoid processes, it is free and borders a hiatus called the tentorial notch, through which the midbrain connects the pons and diencephalon. From the posterior clinoid processes to the internal occipital protuberance, the tentorium cerebelli is attached to bone. Its attachment to the occipital bone encloses the transverse sinuses. Its attachment to the petrous temporal bone, the superior petrosal sinus. The diaphragma celli forms the roof of the sella turcica, the bony fossa surrounding the pituitary gland. A small opening in its center allows the passage of its stalk and vessels. The dura mater of the spinal cord surrounds the cord and extends along the spinal nerves through the intervertebral foramina. It is attached to bone only around the foramen magnum and to the posterior surface of the second and third cervical vertebrae. The spinal dura mater has only an inner meningeal layer. The outer endosteal layer is represented by the periosteum of the vertebral canal. Between the two is the extradural or epidural space. The arachnoid mater, a thin but impermeable membrane, lines the inner surface of the dura mater. It sheathes the brain and spinal cord and envelops the cranial and spinal nerves as far as the foramina. Over the surface of the brain, with the exception of the longitudinal cerebral fissure, the arachnoid mater does not enter but bridges the sulci, covering the cerebral vessels. The arachnoid mater is separated from the dura mater by the subdural space and from the pia mater by the subarachnoid space. In places, the subarachnoid space is expanded to form cisterna. The superior cistern is between the cerebellum and the splenium of the corpus callosum. The interpeduncular cistern between the crura cerebri. The pontine cistern in the basilar groove, and the cerebellomedullary cistern between the cerebellum and the roof of the fourth ventricle. Filled with cerebrospinal fluid, The subarachnoid space contains the larger blood vessels and the cranial nerves, embedded within a fine trabeculae of arachnoid tissue. The subarachnoid space extends along the olfactory nerves to the nose and along the optic nerves to the eyeball. Below, it extends beyond the end of the spinal cord as a small reservoir from which cerebrospinal fluid can be tapped for analysis in a procedure called lumbar puncture. A needle introduced through the interval between the third and fourth lumbar vertebrae pierces the dura and arachnoid mater and enters the subarachnoid space without danger of damage to the spinal cord. Beneath the arachnoid mater is the pia mater, a delicate membrane containing a plexus of minute blood vessels. Over the brain it forms an intimate covering, extending into all the sulci and fissures, and sheathes the cerebral arteries which enter the substance of the brain. Within the ventricles, the pia mater is invaginated and lined by ependyma to form the choroid plexuses. The pia mater of the spinal cord is less vascular than that which covers the brain. It is intimately adherent to the spinal cord, entering the anterior median fissure. The pia mater extends laterally to form the ligamentum denticulatum, which suspends the cord in the dural sac and separates the dorsal and ventral roots. It continues from the end of the cord as the filum terminale. Circulating through the subarachnoid space, the ventricles of the brain and the central canal of the brainstem and spinal cord, is the cerebrospinal fluid. Clear and colorless, with few cells, its total volume is about 130 milliliters. Most of the cerebrospinal fluid is produced by the choroid plexuses in the body and inferior horn of the lateral ventricle and the third and the fourth ventricles. Cerebrospinal fluid flows from the lateral ventricles through the interventricular foramen to the third ventricle, and via the cerebral aqueduct of the midbrain into the fourth ventricle. From the fourth ventricle, the fluid may descend into the central canal of the spinal cord or may enter the subarachnoid space through apertures in the roof of the ventricle. There are three apertures: the foramen of Magendie in the midline and the foramina of Luschka laterally. Within the subarachnoid space, cerebrospinal fluid may descend around the spinal cord or ascend through the cisterns to reach the surface of the cerebrum. Some cerebrospinal fluid re-enters the bloodstream directly through the smaller blood vessels, but most returns through the arachnoid granulations, groups of arachnoid villi which project into the dural venous sinuses, and particularly the superior sagittal sinus. Each villus is an outgrowth of the subarachnoid space which penetrates the dura to fuse with the endothelium of the sinuses. Production of cerebrospinal fluid is continuous, and blockage of its circulation may cause it to accumulate within the ventricles and subarachnoid space, a condition known as hydrocephalus.